Hi, this is for your Chapter 11, Section 3 podcast. If you're looking for Section 2, remember we did that one in class. It was 3 or 4 slides, very short. So if you're looking for that, we did that one in class. This is for Chapter 11, Section 3, Transcription and Translation. Really one whole big topic that's going to be on your EOC and your next test. So it starts with essential dogma theory. Now, proteins are the building blocks for cells and enzymes. So essentially, proteins are the building blocks of life. But how are proteins made? Well, the essential dogma theory states how proteins are made. It says it starts with DNA, the genetic information, then moves to RNA, and then the RNA directs protein synthesis. Now, that's a very simplistic view. I'll be expecting you to know it more in detail, and we'll go over that in class, of course. Now, this sequence of events happen in all organisms, happens from single-cell organisms like bacteria all the way to complex multicellular organisms like humans. And there are 3 types of RNA, which is ribonucleic acid. There are 3 types. There's mRNA, and yes, that is a lowercase m that is written correctly. And the M stands for messenger RNA. We also have a lowercase r, RNA, and that is written correctly as well. That stands for ribosomal RNA. And the last one with a lowercase t RNA, that is written correct as well. That stands for transfer RNA. And we're going to talk about what those actually do. So there are 3 types of RNA. The first one is mRNA, carries the genetic information from the DNA in the nucleus to direct protein synthesis in the cytoplasm, directs how proteins will be made. Why does it go into the cytoplasm? Because that's where ribosomes are located. So it's made a copy of the DNA. So it's going to be a strand here, and it's going to carry a copy of the DNA, and we'll talk about how it does that in a moment. rRNA, scientists have cut us a real break here, associates with protein to form the ribosome. That's why it's called ribosomal RNA, because it makes ribosomes, and ribosomes are where proteins are made. The tRNA transports the amino acids to the ribosome. The mRNA orders the amino acid. The tRNA is going to bring the amino acid that the mRNA ordered and transport it to the ribosome so the amino acids can build the proteins. So RNA, explained a little more, is ribosomal RNA. Type of RNA associates with proteins form ribosomes in the cytoplasm. You can simply summarize it up by saying RNA makes ribosomes, which is where proteins are made. Ribosomes are found in the cytoplasm. So nothing very difficult to understand there if you know about ribosomes, and you should. mRNA is messenger RNA, and mRNA molecules are long strands of RNA nucleotides that are formed complementary to one strand of DNA. Now remember, DNA has A, C, G, and T. RNA has A, C, G, and U instead of that T. So here's how it pairs up. When the mRNA looks at the DNA and it sees an A, it's going to make a complementary strand or a complementary base with a U, because A in DNA will pair with a U in RNA. They're like puzzle pieces that fit perfectly together. And the reason A pairs with U is because we're going from DNA to RNA, specifically mRNA. And RNA doesn't have T, it has U instead of T. C pairs with G, just like it does in DNA. So for every letter C that mRNA sees, it's going to make the complementary base, which is G. When it sees the letter G in DNA, it will make the complementary base C. And when it sees the T in DNA, because remember, DNA has A, C, G, and T, T is going to pair with A. So when you have a T in DNA, the mRNA will use its A to pair with the T. So that's why you see A pairing with U and T pairing with A, because it's going from DNA, which is A, C, G, and T, to mRNA, which is A, C, G, and U. Now you might be saying, I thought RNA didn't have a T, and that's what we were just explaining. It doesn't, but DNA does. When there is a nucleotide T, which stands for thymine, it pairs with A in the RNA. So remember, this right here, for every T that is found in the DNA, mRNA, when it makes a copy, it will pair it with an A. For every A that the DNA has, the mRNA will pair it with a U, because it doesn't have T to pair with the A. So mRNA travels to the nucleus, and then will exit from the nucleus when it's made a copy to the ribosome, where proteins are made. And then tRNA. tRNA is transfer RNA. They're smaller segments of RNA that bring amino acids, which are the building blocks of proteins, to the ribosome. So we have the bases, which are the building blocks of amino acids, and amino acids are the building blocks of protein, and we'll look at that a little more. Now, we're primarily going to be focusing on mRNA and tRNA, because our RNA builds ribosomes. So we need to know what mRNA does, which we just talked about, making a copy of DNA, and what tRNA does. So we talked about transcription and translation. How does that come into play? Well, transcription comes first, and transcription means to make a copy, which means mRNA is going to be involved in transcription. Now, there are two words that are similar. Transcription and translation, very similar. If you're trying to remember which one comes first, if you put them in alphabetical order, transcription would come before translation. Now, transcription is the very first step. The DNA code is transferred to the mRNA inside the nucleus. On the last slide, we just talked about A pairing with U, C pairing with G, G pairing with C, and T pairing with A. That is what we just did, which is essentially transcription. Now, the DNA unwinds, and RNA polymerase reads the DNA strand, making a complementary copy. The mRNA then takes the copied code to the cytoplasm, where the ribosomes are. Now, mRNA is significantly shorter than DNA code. Scientists found this out and said there are introns and exons. So what are these? And don't be misled by the prefix. So the introns are removed from the mRNA code. The exons are included in the final mRNA code. And yes, you read that right. Introns are removed. Exons are included in the final mRNA code. Don't be misled by the prefix N or the prefix X here. They're very easily confused. It's a very easy one to study, but if you don't study it, you're going to be misled by the N and X prefix, and you'll get it wrong, and we don't want that happening. So let's talk about what codons are. Now, scientists knew that DNA coded for proteins, but DNA only has 4 bases. There are no more, there are no less, there's no hidden bases, there's no extra here. It's just simply A, C, G, and T. The only difference in organisms was the order of the bases. They needed to figure out the amount of bases, whether it's the A, C, G, T, those are the bases, the nucleotides we're talking about. They wanted to know how many bases code for an amino acid, because there's billions of letters, but how many letters in a row will actually code for an amino acid? So in the 1960s, they found out that it's a 3-letter code. So every 3 letters of DNA, ACC, CAC, CGG, any combination that you can think of, will code for a specific amino acid. So this means that every set of 3 letters code for 1 amino acid, which we just said. With 4 letters and a little math, you can work out that there are a possible 64 combinations. There are no more, there are no less. Now, some combinations of amino acids code for the same thing. We'll take a look at how to read a chart and do some practice in class. We'll also go over it briefly in this podcast. When you're talking about codons, you're talking about going from DNA to mRNA. So anytime codons are mentioned, you're talking about DNA going to mRNA. Now, mRNA to tRNA, they're not called codons, they're called something else, and we'll talk about that in a moment. So here's our chart. So we're going to use the example UCC and GGG. This has already been transferred to mRNA for us in this example. The starting DNA was AGG. I know A pairs with U, G pairs with C, G pairs with C. I have my 3 letters. So here's our circle chart. It can look a little overwhelming, but it's very, very easy to follow. You start from the inside out. So my first letter is U. Here's my U right here. Now I'm looking at one of these 4 letters. My next letter is C. So I'm going to go from U to C. Now I have one more letter left, which is C. But it doesn't matter what that 3rd letter is. It codes for this right here, serine. And then I have GGG. Start from the inside out. I have G. Here's my other G. Doesn't really matter if it's a G, A, C, or U, because they all code for the same thing, glycine. Now let's talk about methionine and our stop codons. Methionine is our start codon. This says, hey, we're going to start making a protein. So protein's usually going to start with A, U, and G. That says, hey, we're making a protein. Let's start making it right now. So that will tell the ribosome, okay, we've stopped making one. Let's start on the next one. We also have the stop codon, which is UAA, UAG, and it's also right here, U, G, and A, and that says, hey, stop making the protein. We're done with this one. It's time to move on to the next. So after we've done transcription, we need to do translation. So once the mRNA enters the cytoplasm, it's going to make its way to the ribosome, and it's going to attach to that ribosome. The mRNA code is read and translated to tRNA molecules, and this is called translation. So translation is going from mRNA to tRNA. Well, what about that rRNA? Where does that come in? Well, remember, RNA helps make the organelle ribosomes, so it's not involved in transcription or translation. The tRNA molecules make a complementary copy of the mRNA and bring the amino acid coded for by the mRNA. It transfers the amino acid, which is why it's called transfer RNA, and the amino acids will be the building blocks of proteins. So mRNA orders it, the tRNA reads the order, and then we'll bring it over. So here's how it pairs up. Now we're going from mRNA to tRNA, so we only have the letters A, C, G, and U. RNA doesn't have T, so A is going to pair with U, C is going to pair with G, G is going to pair with C, and U is going to pair with A, because we're going from mRNA to tRNA, and RNA doesn't have T, it has U's. So yes, it's that simple. Notice the T's have been replaced with U's because RNA does not have T's. The three complementary bases to mRNA are called anti-codons. Remember, we said DNA to mRNA, when mRNA is making that complementary code and letters of three bases long, those are called codons. When you're going from mRNA to tRNA, making three-letter complementary bases, we call those anti-codons. Codon and anti-codons pair up. So here we have translation. And I know this is translation because I see the letter U. I don't see DNA here. If I saw DNA, and going from DNA to mRNA, I would know we'd be in transcription. But because I don't see any T's here, I know we're dealing strictly with mRNA and tRNA, which is why we're doing translation. So here's my mRNA. It's made a complementary copy of the DNA. And then here we see we have GUC. What does G pair up with? G pairs up with C. U pairs up with A, and C pairs up with G. So if we're looking for this, it's in here somewhere. We know that G pairs up with C. Here's a C. We're looking for CA something. So here it is, CAG. This is what it pairs up with right here. The G, no, that's not right. The G pairs up with C, the U pairs up with A, the C pairs up with G. So it'd be coding for this amino acid right here. So then once it drops off the amino acid, and that are these things right here on the top, it will go and figure out what it needs to bring next. So the tRNA brings in the amino acid, and these are these little bubble circles at the top. This is the codons, which pairs with the anti-codons. So that's all there is to it. We can see this one right here. U is going to pair with A, G is going to pair with C, and A is going to pair with U. That is the anti-codon pairing up with codon. So anti-codon is when with tRNA, the codons deal with DNA to mRNA. So here are my codons, which are going to pair up with my anti-codons. They're going to drop the amino acid. tRNA is going to get the next one and get the next amino acid ready. And the amino acids together are going to build the proteins. So here's an example of a sequence. So for simplicity, we'll run through two examples. There are billions of letters. We don't have the time to run through billions of letters. If you have to run through a billion letters to figure this out, I don't know what to tell you. So imagine we have two codons, ATA, CGA. So we know right away the two codons are DNA because we use the word codon, and we also see a letter T in the first codon. And a codon is just is three bases. That's it, because codons or DNA is read three letters at a time from left to right. So this will read ATA, which will code for an amino acid, and then CGA, which will code for another amino acid. So the first step is transcription, which is DNA to mRNA. We got to use those pairing rules. So we have ATA. What does A pair with? I know that A is going to pair with U. I know T is going to pair with A, and I know A is going to pair with U. So mRNA will make a copy of ATA, and it makes those complementary bases. That's how we get UAU. And next is translation. And this is when we're going from mRNA to tRNA. So once you have UAU, we can use the chart to figure out what tRNA is bringing, and we can do that on the next slide. So we have U pairs up with A pairs up with U pairs up with A. So we have to use mRNA when we use this chart. And when I look back, I see it's UAU. I start in the middle here. I start at U. My next letter is A, which is right here. Now I'm looking for one of these 4 letters. The last letter was U. And right here, I have what I am looking for, which is tyrosine. So here's the next one for CGA. DNA to mRNA, that is transcription. That comes first. So I have CGA. I use the pairing rules. What does C pair with? C pairs with G. What does G pair with? That pairs with C. What does A pair with when I'm going from DNA to mRNA? A pairs with U. So that's how I got those 3 letters. We can then figure out what is the tRNA will bring. So we can go back to the chart real quick. We have GCU. So we start here, G. Look for the next letter. Here's my C. Doesn't really matter what the last letter is, but U. And I have alanine. So mRNA to tRNA, which is translation. The codon of the mRNA will pair up with the anti-codon of the tRNA. So my mRNA was GCU. I know G pairs with C. C pairs with G. And the U, when going to mRNA to tRNA, is going to pair with A. All the amino acids in the mRNA sequence will make the protein. Now, we just ran through 2 sequences really quick for simplicity. Now, it's important to note that the mRNA section goes all at one time. The code is read 3 letters at a time, which we call codons, and it's always read 3 letters at a time from left to right. The codons pair up with the anti-codons, which is the tRNA, and that is the central dogma theory. DNA to RNA to proteins. If you have any questions, please write them down and leave a comment below. We'll see you guys in class.